Okay, great. Just we're a couple minutes after the hour, so let's go ahead and get started for the sake of time and make sure that both our presenters have plenty of time to present their research. I wanted to thank you all for joining today's ASBMR Member Spotlight. Welcome back if you've attended one before, and welcome if you're new. The series is brought to you by the ASBMR Diversity, Equity, and Inclusion Committee, and it aims to highlight the research of ASBMR members within a collaborative setting, and aims in particular to shine a light on the research being done by ASBMR members belonging to groups who are historically underrepresented in biomedical research. Today's spotlight will have two presenters. First, we have Dr. Baohong Zhao, Associate Professor at the Hospital for Special Surgery, who will present on type 1 IFN signaling and osteoblasts and osteogenesis. Second, our presenter will be Marta Galan-Diaz, Associate Research Scientist at Columbia University, who will present on analyzing the role of bone marrow microenvironment in acute myeloid leukemia progression. With that, don't want to waste any time. I'd like to go ahead and invite Dr. Zhao to go ahead and begin her presentation. And just remind all attendees to please keep yourself on mute. Following each presentation, there will be time for Q&A with the audience. So at that point, you can feel free to unmute yourself to ask a question, or you can always use the chat feature to ask a question that way. So Dr. Zhao, please feel free to go ahead.
Here we go. Let me first share screen. See. Can you see my slide?
We can hear your screen, or we can see your screen, but your audio is a little quiet.
Oh, little quiet. Okay, let me increase. Let me increase. Oh, can you see my screen now?
Yes.
Okay. So let me increase the one. Is it better now?
I can hear you better, yeah.
Okay, so now it's better. Okay, so first I would like to thank ASBMR, especially the Diversity Committee, to really initiate this event. It's very helpful and a very good opportunity for all of our researchers from all over the world to start to gather together on the Zoom meeting and then to share the recent research. So this is my really deep appreciation for this chance. So let me okay, so today I'm going to talk about a relatively new direction in my lab. So it's about endogenous type 1 interferon signaling in osteoblasts and osteogenesis. So, you know, as we know, actually most of our cells in our human body, I think most of cells are very competitive to response to type 1 interferon, including interferon alpha and interferon beta, especially in response to virus infection. But most studies are focused on the immune cells so far. For example, macrophages study how they respond to interferon and the toll-like receptor, the virus in the infection and the inflammation. So the studies for the type 1 interferon in osteoblasts and osteogenesis, especially the endogenous type 1 interferon, that means autocrine secreted, osteoblast secreted interferon beta, they have autocrine effects on themselves, are still much underappreciated. So I would like to, talk about this topic in osteoblast. So first, I would like to briefly introduce some background. So to let you know how we started this research direction. So how we found this kind of project. So it started with actually the project when we searched for the novel regulator that regulate bone remodeling. And then, I will start from here to introduce the whole story. So For the bone biologists in the field, it is well known that the bone homeostasis is maintained by a balance between the osteoclast-mediated bone resorption and the osteoblast-mediated bone formation. So this is a pretty well-established model. And then there are a bunch of canonical coupling factors that couple these two bone cell types that can work together nicely to maintain the bone volume, including, for example, BMP 6S1P, and then the very classical run ligand OPG access, etc. But recently, we found actually the bone remodeling is also controlled by a certain regulators that are not limited to the canonical coupling factors that are secreted by the cells. So these regulators could be transcription factor, could be any proteins in the cells, but they simultaneously control the differentiation and the activity of both all three, also including osteocyte, the bone cell types. So we think about this non-canonical bone remodeling regulator can expand our understanding of the mechanism of bone remodeling. So from there, we identify this axis, the novel axis called DAP6 interferon beta. And then we found this axis can regulate, can do this thing, simultaneous control both cell type in the bone remodeling. So what is DAP6? So here I will start to briefly discuss. So DAP6 is originally an immunoregulator that is extensively studied in immunology field, such as especially in T cells. So, and then recently, especially there are a bunch of studies here, I just listed the major one that found the DAP6 function in the innate adaptive immunity as well in the autoimmunity, such as in the human diseases, lupus and rheumatoid arthritis. So further emphasize the importance of DAP6 in the human diseases. So what really brought DAP6 in my eyes into the spotlight? So that's started with a recent research. They found they used the GWAS and meta-analysis of bone mineral density, identified the DAP6. This is the full name if you are interested to know that originally they identified. So they identified DAP6 as a novel loci associated with BMD, importantly at different skeleton sites. So this is a very promising and important regulator when we think about that. But how DAP6 regulates the BMD and bone homeostasis was not unknown at the time. This is only GWAS study. And then another piece of evidence that draw us to this spotlight is the DAP6 deficient mice crossed with T cell receptor transgenic mice develop rheumatoid arthritis the symptoms with bone erosion. So all of this piece of evidence led us to, okay, let's see whether DAP6 has a role in the bone homeostasis. So here I show you the basic structure, what basically a DAP6 molecular structure looks like. It has a very unique molecular structure. From here, you can see the N-terminal. There is a EF10 that has potential calcium binding ability, followed by a pH domain. Is a PIP3 binding domain. And then on the C-terminal, there is a double homology domain. This domain actually exhibits GF rho family guanine nucleotide exchange factor activity at the C terminals. So this is interesting. This structure is not similar to the classical GF structure. In the classical GF structure, this DH domain is on the N terminals, while DAP6 is on the C terminals. So you'll see the studies in the field. In addition to the GF activity of DAP6, you will see DAP6 actually plays a multiple function role to activate the downstream factors. For example, this model shows DAP6 signaling in T cells. You can see in addition to this DAP6 under the T cell receptor that activate the downstream RAC CDC42 to activate certain effector regulators. But on the other hand, for the more well-known, DAP6 interacts with RF4 they interact together to regulate inflammatory cytokine IL-17 and IL-21. So these actions are very important. Recently, in the autoimmunity, human autoimmunity disease has, and the lupus and RA has demonstrated that the axis is pretty active and present in those diseases. So here I give you overall view of DAF6 so far in the immunology field. So it really could have multiple functions, not limited to the GF activity. So what's the role of DAF6 in bone remodeling? And then we want to know what are the underlying mechanisms. So first we look at the bone phenotype. So this is the global knockout. So you can see in the DAF6 knockout mice, you can see the reduced bone volume. Actually, this DAF6 is also, has osteoporotic bone phenotype with enhanced osteoclast differentiation. And we published these results a couple of years ago, and we found the mechanism is like I draw in this model. The DAF6 activate type I interferon response, especially interferon beta. We know in the macrophage osteoblast differentiation process is major interferon beta. And then use this autocrine feedback inhibition by the interferon beta to inhibit osteoblast differentiation, the bone resorption, also including in the inflammatory bone resorption. So here, this is a novel finding in osteoclast. Type 1 feedback inhibition by type 1 interferon beta in osteoclast is well established, I think by two decades already. So this is not surprising because osteoclasts derived from macrophages can secrete a little amount of interferon beta. So we're wondering, what's the role of DAF6 in osteoblast, the bone formation part, because this is a global knockout mice. So we started to first test the expression. We saw first during the osteoblast differentiation when we use the cavarian osteoblast culture in vitro. This is the well-established cultural system that is used by almost all of the labs in the world. So here you can see the DAF6 expression is gradually induced during the differentiation at both mRNA level and the protein level. And furthermore, on the bone slices, this is in vivo. So you can see this part, the black part is the bone, and on the surface you can see a layer of the cells. They are osteoblasts. The green staining shows the DAF6 molecules. So the arrows you can clearly show these osteoblasts express DAF6 in vivo. So okay, so we know first DAF6 expression there exists in the cells, and we studied their function. So we took advantage of the DAF6 knockout cells, the cavarian from the cavarian bones. So here you can see the osteoblast differentiation indicated by LP staining, red staining, a bunch of osteoblast marker gene expressions such as ALP, BSP, osteocalcin was significantly enhanced in the DAF6 knockout cell cultures, indicating that DAF6 functions as inhibitor in osteoblast differentiation. This data already just published in the eLab. So now we want to know the DAF6 function in vivo. So first we perform double labeling of new bone formation. So here you can see the slices of the bones from wild type and the DAF6 knockout mice. They are calcium labeled, double labeled, so you can see the gap distance here, right? DAF6 knockout mice show significantly enhanced bone formation rate than the wild type mice. And then further down, we analyze a bunch of osteoblast parameters to see the osteoblast activity and function. So here is you see this the staining. So the green shows the bone part, and then the red are bone marrows. So on the surface of bones, you can see the layer of osteoblast. In the KO, you can see actually the osteoblast cells are bigger and more when we do the quantification than the wild type cells, including osteoblast surface. It's very clearly, you can show the data you can appreciate the data. They show clearly enhanced in the DAP6 knockout mice. So overall, this data demonstrate that DAP6 is an inhibitor both in vitro and in vivo for the osteoblast differentiation, osteogenesis, and bone formation. So then we want to know what are the underlying mechanisms in osteoblast, right? So this never studied for the DAP6 molecule. So we do first the RNA sequencing analysis using the cavernous osteoblast isolated from the wild type and the knockout cells. This is the volcano plot. You can see the differentially expressed genes. They are expressed. Some are high, especially in wild type. Some are especially high in DAP6. So when we do pathway analysis, it's very clear to show us if you can appreciate it here. The top is the pathway that are enriched in wild type. You can see on the top enriched pathway is type 1 interferon signaling. And of course, there are some other interferon signaling pathways were also enriched. In contrast, in the DAP6 knockout cell cultures, you can see the enriched pathways are endochondral ossification, osteoblasts. So these pathway analyses are pretty consistent with the phenotype we just saw, right? So in the DAP6 knockout, there are the osteoblasts and the bone formation phenotype were significantly enhanced. So this draw us pretty strong interest. So these are osteoblasts, right? Mesenchymal lineage cells, but they show pretty significant enrichment of type 1 interferon signaling. So there's quite a few questions. So what are the source of this type 1 interferon? These are the osteoblast culture. We remove the endotoxin, any contamination, we use the control. There's no toll-like receptor potential contamination in these cultures. And also we did, I didn't show the data here, but in the paper we show that. So between the wild type and DAP6 cell culture, in this culture system, there are a little bit, bone marrow cells, but they are identical. So, but very striking, wild type has a strong interferon signaling enrichment. So we further look at the details. So here, you see this panel, we show the heat maps. So the left heat map show the interferon stimulated genes. They are target genes, responsive genes, specifically response to type 1 interferon, very specific, this bunch of genes. Here very clearly, number 1, number 2 means two biological replicates of the RNA sequencing experiments. So during the differentiation, we compare the day 0 and the day 12. This is the cavernous osteoblast cultured with beta-glycerophosphate and vitamin C. So this is a common culture system. So you can see in wild type, a bunch of these interferon stimulated genes, we just call it ISG genes. So these ISGs are significantly enhanced compared with DAP6 knockout cell cultures. In contrast, we also look at the osteoblast genes, marker genes especially, and then you can see in the DAP6 knockout cultures, this gene expression was significantly higher than the wild type cell culture. And the GC enrichment analysis further support this data. So in wild type, basically the type 1 interferon responsive genes are highly enriched, while in the DAP6 knockout, osteoblast genes were highly enriched. So these two cell cultures have a very, very clear and distinct enriched pathway. So the molecular basis is clear here. So the bottom, we just you perform some RT-PCRs to further confirm and demonstrate the ISG gene expression that show up during the osteoblast differentiation, the white bars. So these are a bunch of very classical ISG genes in osteoblasts. Generally, these genes, you see the regulation of these genes are in the macrophage immune cells. But here in osteoblasts, we saw clearly induction. You can see the clear induction of the PCR. While in the DAP6, all of these gene expression were inhibited. Well, so this is the point I want to tell you, starting from the original, the story. So from here, we really feel, okay, the type 1 interferon response in osteoblasts may have some roles. So this is the whole story, how started, we see it from my presentation title. And then, a lot of things, whether there is a function, right, or not, you have this gene expression, but whether they're functional or not is another question. So first we use, okay, let's use blocking antibody. This is already interferon blocking antibody. This is already demonstrated, very effective and specific. Here you can see this, the upper panel, we tested the ISG genes, MX1, MK2. And here you can see what in the wild type cells represented by the white bars, they're induced. But when you give the interferon beta blocking antibody, it significantly decrease the induction of ISGs in wild type cells. Well, in the DAP6, because the original level is pretty low, but this blocking antibody can further bring the expression level down. On the other hand, for the osteoblast marker genes, BSB, osteocalcin, you can see after blocking the third column, right? After blocking the cell cultures with interferon beta blocking antibody, you can see that will bring the marker gene expression enhanced in the wild type, and to a little bit less extent to in the KO, because KO starting point is pretty high already. And then the allogeneic red staining also showed a similar phenotype. So blocking antibody of type 1 interferon enhanced the mineralization. So furthermore, we tested blocking antibody, demonstrated there is some endogenous interferon beta that is present in the osteoblast cultures. They may be, we were talking about, because we already excluded whatever the potential brought bring in by the humans, by the researchers. So there is a possibility, highly likely possibility. I will show the data later. Before that, we further tested the cellular response of this osteoblast to type 1 interferon. So we give a very little amount interferon beta to try to mimic endogenous level of interferon beta. So here we tested the downstream signaling indicated by the phosphorus study 1, phosphorus study 3. And then here you see very little amount. This is 1 nanogram per ml. I think in the wild type, you can see clearly induction of this phosphorus study 1 and the phosphorus study in wild type osteoblast. That means osteoblast has very nice response to type 1 interferon. And then in the DAP6 knockout, you can see the signaling pathway were much weaker compared than those in the wild type cells. And then the IS gene, the downstream IS gene showed a similar pattern, right? And then when we gave a dose-dependent amount of the interferon beta, here 10 unit, very low dose, and a little bit higher, 100 unit per ml. So you see wild type, we clearly inhibit osteoblast, while in DAP6 knockout, is very minimal inhibition. So here is, that means, DAP6 also regulates cellular response of this osteoblast to interferon beta. So this is another point, this regulatory responses. So here, just view this end, give you a model. So we found actually a shared pathway mediated by the DAP6 interferon that suppresses the differentiation of both osteoclasts and the osteoblasts. So that's why we regard this DAP6 as a non-canonical novel bone remodeling regulator. So if we remove this DAP6 by either genetic knockout or by disease condition, for example, the GWAS show some people may have this kind of defects or the functional defects of these genes. So interferon response will be suppressed in osteoblasts. So the results will increase both the osteoclast mediated bone resorption and increase osteoblast mediated bone formation. but the net result is osteoporosis. So this step 6 actually regulates a high turnover osteoporosis in the bone homeostasis. So here we back to the question I left a few slides before. So because we use blocking antibody, right? So whether this hypothesis there is secreted, there is endogenous interferon beta in the cells, is that true? So and then we tested interferon beta level in the mouse serum. So yeah, this is pretty nice. So you can see the wild type, they have a low amount, but they have amount of this interferon beta, and then in the DAP6 has a lower interferon serum level. And in the cell culture, you can see clearly during the differentiation of calvarial osteoblasts. So you can see the interferon beta protein is also enhanced during differentiation and then maintained at a certain level. Well, in the DAP6 knockout, you can really marginally at you will see this level, so almost nothing at the interferon beta. So this the DAP6 is really an important regulator for the interferon beta expression in the osteoblast differentiation culture. So and then we ask whether this type of interferon has a function or not, right? So still the question. They regulate ISG, they're present in the cell culture. So we use the type 1 interferon receptor knockout mice, and then we performed osteoclast differentiation. So by LP staining, arsenide staining, and a bunch of these gene expression, including osteoblast marker genes, ISG genes, ISG gene expression as a parallel demonstration of this type 1 interferon. So they are absent in the knockout, the black bar in the interferon receptor knockout because our receptor is knockout. So you can see actually the lack of type 1 interferon signaling in osteoblast significantly promote osteoclastogenesis. So that's support our hypothesis. So interferon beta is secreted by osteoblasts during differentiation, and the interferon beta play an important inhibitory role in the osteoblast differentiation. And this interferon beta expression and the downstream ISG gene expression is regulated by an upstream regulator DAP6. So this is so far enough. Then we further I took a few minutes more. We'll finish. I know it's already 1:30. So we know, this is a question. I just took 5 minutes more. So we know, but how DAP6 regulates, right? Regulates ISG, regulates this whole bunch of biological events. So we found another molecular regulator called PKR. The PKR is protein kinase R. So this is originally identified is also in the type 1 interferon pathway in response to virus. So generally, this regulates the translation of the cells when they see the viral infection. So this one also the downstream in the T cell in the macrophage. So the downstream they can regulate partially interferon beta expression. That's why we think about recheck PPR. And in our osteoclast study, we published a paper in Nature Communication a couple of years ago. We found this PPR is a regulator for tapping the interferon beta expression in osteoclast. So that's why we test whether the PKR is also in osteoclast. Very interesting, you'll see, yeah, the DAP6 regulates PKR expression. And then when we further silence the PKR in the osteoblast differentiation by knocking down this molecule, so we test the effects, the knocking down effects on the differentiation. So you can see this is very clearly. So in osteoblast, if you knock down the PKR, you see the type 1, the interferon beta expression is drastically reduced. And then following that, the downstream ISG gene expression will further reduced in the knockdown condition. Well, in contrast, the osteoblast differentiation is promoted and by the LP staining at a bunch of osteoclast markers. So further, after the knocking down, we further want to overexpress because in the DAP6 knockout, PKR expression is low, it's pretty low. So we give back to this PKR, and then we found when we give back the PKR in the DAP6 knockout, we see we can saw this interferon expression is restored, is enhanced, and followed by the enhancement of the expression of a bunch of ASG genes. On the other hand, the osteomarker genes were reduced. So far, this DAP6 deficiency inhibits endogenous expression of interferon beta and the downstream ISG gene expression through the downregulation of PKR in osteoblasts. This is what we the downstream mechanism part. So these key points is basically here. So we know PKR is a well-established immunoregulator in T cells, and here we found DAP6 also as a novel bone remodeling regulator. So this is non-canonical novel regulator that can regulate both osteoclast differentiation and osteoblast differentiation simultaneously. So we regard this as a novel bone remodeling regulator because we do pretty simultaneous effects. And then also this we establish a pathway access here. So DAP6 works through the PKR and induce the interferon beta endogenous expression and induce the downstream type 1 interferon response. By this access, DAP6 inhibits the osteoblast differentiation and the bone formation. On the other hand, very importantly, we feel this is project started with DAP6, but we found actually the ISG, this interferon stimulated gene expression, is generally induced along osteoclast differentiation, but it is reversely always not only limited to wild type DAP6 in other cell osteoblast culture also show reversely correlated with osteogenic potential of the cell culture. So we feel this ISG gene expression could be a marker, molecular marker to indicate the osteoblast differentiation potential. Okay, thanks for your attention. I just briefly, really show my thanks to all of my lab members, especially Christine Miller, Kazuki Inoue, Zhonghao Deng, Courtney Ng. So they contributed to these projects, and also my research support from this wonderful agencies. And also, a quick, I have a postdoc openings here available. If anyone interested, please just contact me directly. Thanks very much for your attention. I took a little bit more time. Sorry about that. I welcome any question. If not, later is okay. Thank you.
Yeah, thank you, Dr. Zhao. If you don't mind just stopping to share your screen. And then I think just in the interest of time, why don't we go ahead and start with our second presentation, and then at that point at the end, we can take questions for both presenters. Sure. Just want to make sure that Dr. Khan has plenty of time. If that sounds okay with everyone, please, Marta, feel free to go ahead and begin your presentation.
Sure.